let me welcome you to our final machine learning algorithm talk of the fall semester. We'll have a few more in the spring, but today we're really very grateful to have Professor Jon Steingrimsson, who's in the Department of Biostatistics at Brown, as a speaker, talking to us about how we evaluate the machine learning algorithms that we've been talking about over the last couple of talks. Professor Steingrimsson works on developing machine learning algorithms for health data and on applications of machine learning to health data. This includes developing algorithms for time to event data, for imaging analysis, estimation of personalized treatment effects, and for discovering subgroups with differential treatment effects. His collaborative work primarily focuses on imaging for cancer. And so with that, let me turn it over to Jon. And if you can put any questions in the chat, I will monitor those, and we will have 15 minutes at the end for Q&A.
Yeah, thank you, Chris, and thank you to the organizers for having me here, and thank you all for attending. So this is supposed to be a very high-level talk that doesn't require a lot of background in machine learning or math or anything like that. So I'm hoping this should be accessible to people without those backgrounds, and it might even be too elementary for some of you. So yeah, here's my email. I'm happy to answer questions, obviously, during the talk, after the talk, and even after the session ends through email. So the focus here is on evaluation of prediction models. And so as you know, some of you might have gone to Karsten's talk, prediction models sort of take as an input patient characteristics and provide a prediction for an outcome. Obviously, this is more general. It doesn't have to be patient characteristics, but in health, that is pretty common. So two sort of examples would be, you're interested in sort of knowing the 2-year risk of developing breast cancer, and you're going to base that prediction on data from mammography images, either using the images themselves or features extracted from those images, and then you're also going to sort of add to that some standard clinical risk factors. And another thing that you see pretty frequently is sort of a risk prediction model for cardiovascular disease, like the Framingham risk score and other things, where you might be interested in predicting sort of, say, 10-year probability of major cardiovascular events, and you're basing that on some clinical data. So, but the key thing is you have some inputs into the models, which usually are patient characteristics like images, clinical factors, and so on. And based on those inputs, you're trying to make some prediction about an outcome. And the objective here is not on, or the focus here is on evaluation of machine learning algorithms. We are not going to be focusing on how you build these machine learning algorithms, how you develop them, or so on. We are basically assuming you have an algorithm, and we want to answer the question of how well does it do. And this is even more general than a machine learning algorithm. You could think about like a biomarker. It's not an algorithm, like PSA levels, BI-RADS score from medical images, and so on. This also, everything we are going to be saying, almost everything we are going to be saying also applies to that. Essentially, all you need is some kind of a process, whether it's an algorithm or a marker, that gives you some information or prediction about an outcome. And then the question is, how well does that process predict the outcome? And that's basically what we are going to be focusing on here. So we have a machine learning algorithm. How well does it do? And one concept that is important to the idea of evaluation of an algorithm is that to avoid over-optimistic performance, no data point should be used to both fit the model and evaluate it. So there should be complete separation between the data points or observations that are used to fit the model or build the model or train the algorithms and the models used to evaluate it. And the reason is because if there is some sort of if there is some common point used in both, then you're going to get over-optimistic performances. And one sort of aspect of that it comes to the concept of overfitting. So in this figure here, you have 5 data points indicated by these black dots. and then you have a prediction model for the expected value of the outcome condition on the covariate, which is given by that blue line here. And this would be called the training set, because this is the data that was used to build the model. And you can see that the model goes very sort of, it fits too snugly in the sense that it goes through all of the data points very closely. So it's fitting the training data very well here. And if you would evaluate performance on this data set, and if we say performance is evaluated by the distance from the data point to the prediction, where the prediction is given by the blue line, you could see for the first data point here, they are just the same. So the prediction error for this data point is 0. They're just, that prediction is perfect for that data point. And same for the second data point. But the reason for that is overfitting, because the data or the algorithm is tailored too much to the data. For example, a simple line would work pretty well here. And say you had a new data set where the data point, instead of being here, was here. Then you're getting too big, like you're getting a pretty substantial difference. So the basic takeaway point here is, the important takeaway point here is that no data point should be used for both fit the model and evaluate it. Otherwise, you're getting over-optimistic performance and you're getting biased evaluation measures. And another important concept is sort of what we call internal versus external validation. And for some of you who have been sort of working in clinical trials, this is often called internal versus external validity in the context of clinical trials. So internal validation refers to estimating the performance on the same data set as was used to develop the algorithm or the marker. And here we say the same data set, but we still are doing it on different data points. So there's a difference there. And that can be done in several ways. Two common ways to do that are training test set split and cross-validation. I will go into details about what that means later. But the key thing here is that the same data set is used to build the model as well as evaluate it. And then you have the concept of external validation. There are different data set where you're evaluating the algorithm or the model on a different data set than it was used to develop the algorithm. So you could think about like the algorithm being developed in a data set from the UK, and then you want to say, how well does that algorithm do in the US? That would be an example of an internal validation. No, an external validation, sorry, where the algorithm is fit in one setting and then evaluated in a different setting, or in a different data set, basically. Okay, so two types of internal validation were sort of a test and a train split and cross-validation. And here the key sort of concept of both of these is to avoid this concept of over-optimism, where the same data point is used both to fit and evaluate the model. So this is illustrated here in this figure here, where you have by the green rectangle here, you have a data set, and now you split that data set into two pieces. Where the first piece is what we call the training set, it's given by the blue box here, and the second piece is what we call the test set, which is denoted by the red piece here, or the red box here. And note that these two sets are disjoint, meaning that no single observation is in both of these data sets. And the idea here is that you would build your model or train your algorithm on the training set, and then you would evaluate how well it does on the test set. And this is an example of an internal validation, because this is done on the same data set. But this is sort of a valid way of doing internal validation, because there is no overlap between the training and the test set. And you can even make this a little more complex. In many settings and in many machine learning algorithms, you have a lot of there's a lot of flexibility. There is a lot of what we call tuning parameters that you need to select. Like in deep learning, you might need to select how many layers are you going to be using. And even in simpler models like linear regression, you might be saying, which interaction should I use? Which functional form of a covariate should I use? Should I use a log transformation? Should I square that covariate and so on? So there's a lot of choices you make through sort of the development process. And one way of selecting which model to choose as your final model would be to use a validation set. So you build the model on the training set, and then you estimate it on the validation set, and then you sort of redo that process for multiple models. And the final model selected would be the one that gives you the highest sort of prediction accuracy or highest or best measure of model performance on the validation set. And that becomes your final model. And now you want to do this internal validation, then you would apply that model to the test set. So that means the test set was never used in this process. It was never used in the training process, and it was also never used to select among those many models which one should we select. So that completely avoids any over-optimism in your estimation of model performance or evaluation of well your algorithm is doing. A related idea, a slightly more complex but a very related idea, is the idea of cross-validation. But now, instead of splitting the data only into two parts, a training and a test set, you split it into, say, 5 parts, which is what is done in this figure here. So we have we split the data into 5 parts, and then on all but one of them, we train the model, and then we evaluate it on the last part. So in this third, and then you repeat that process for every single part being the one that's being evaluated on, and that gives you 5 measures of model performance associated with each of the iteration, and then you would average that measure of model performance across those iterations. So this is illustrated in the figure here, where in the first iteration, again, you have 5 parts. The first 4 parts are going to be your training set, where you train the model, and the last part is going to be your test set. And when you do that, you get some sort of error out of that. And then you repeat the process just with a different part of the data being the test set. So in iteration 2, the fourth part of the data would be the test set. And then in iteration 3, the third, and so on. And repeating that over all 5 iterations gives you 5 errors, error 1 to error 5. And then the sort of cross-validated error would be the average across those 5 errors. And the advantage of cross-validation compared to just a single train and a test set split would be it's a little more efficient in the sense that each part of the data both serves as a test set and as a training set in that process, just depending on what iteration you are in. Like the 5th part serves as a test set in iteration 1, but as a training set in all other iterations. So it's slightly more efficient, or can be even substantially more efficient depending on the setting, than this single test and train set splitting. So those are 2 methods of doing internal validation. And these are pretty agnostic to how you're going to do it, what measure of model performance you're going to be using. And another thing that I personally think is pretty important, but this is maybe less mentioned in the literature, is the distinction between prediction versus diagnosis. So diagnosis or detection is basically asking the question about the current time. Is there cancer? Does this patient have cancer now? Looking at this MRI, does this participant have breast cancer? That's a question about the current status. It's a detection problem, a diagnosis problem. And then there is the question about prediction, which is trying to say something about what happens in the future. Like predicting whether, based on your current status, what is the likelihood of you developing breast cancer within the next year? So the key distinction is one is saying something about what's happening now. The other one is saying something about what's going to happen in the future. Okay. Switching gears a little bit. So all of the things, so just to summarize what we have done so far. We started with this concept of overfitting, and the main takeaway point there was basically it's important to ensure that the model you're building is built or fitted or trained on a different data set than the data set you use to evaluate it. That's a key consideration when you want to avoid having over-optimistic model performance. So it's a way to get unbiased estimators of model performance. Then there are 2 ways of doing validation. There's internal validation, which is probably more common. That's sort of the first step you would do, which is basically evaluating the model on the same data set as was used to develop the model. And that can be done either through training and test set splitting or cross-validation. And if the model shows good performance using internal validation, a very standard question that sort of regulatory agencies, for example, the FDA would require you to do would be to say, okay, it worked well in that setting. But what about if you transfer that model or use that model in a different setting that might have a different set of patients, a different sort of case mix of participants, different cohort distribution, different racial and ethnic distribution, and so on? How would it do? And that's what we would call external validation, where you're validating the model on a different data set than was used to develop the algorithm. And like I said before, test and train are versions of internal validation, and the same can be said about cross-validation. And then this concept of prediction versus diagnosis. The diagnosis or detection means detecting whether it's here now. Prediction means saying something about the future. And all of what I've said so far is pretty agnostic to the measure of model performance. We'll be using AUC, sensitivity, MSE, specificity, whatever. Like, you should still be using like valid measure that don't use the same data points to train and test. You would also, if you want internal validation, you could use that with the test and training split or cross-validation. Prediction and diagnosis is a little more goes a little more into the question which measures are appropriate. In my opinion, different measures are appropriate for diagnosis than prediction. But so far, we haven't touched upon the measures or the methods of evaluation. And what the appropriate methods are depends on the type of outcome. And we are going to be talking mostly about binary outcome, but we'll briefly touch base on both continuous and time to event outcomes. And by a binary outcome, we mean can only take two values, like presence or absence of disease progression. By continuous, we mean, and this is probably a lot of you have heard this before, can take quote unquote all values at least within a specific range, like height or viral load and something like that. And another outcome that is very popular in medical research is time to event, like overall time to overall survival, time to death, disease-free disease-free survival, progression-free survival, event-free survival, viral time until viral load suppression, and so on. And these prediction model built for these type of outcomes tend to be harder to evaluate because of what we call right censoring, which many of you might have heard of. Okay, so binary outcome. How do we go about evaluating a model for a binary outcome? And here we are going to sort of we are going to use three different type of measures, which I here call class predictions, which you're going to sort of say sensitivity, specificity, negative and positive predictive value, and accuracy would fall in that category. Sort of how well are you doing at predicting sort of diseased versus non-diseased, or within each class in some sense. I'll define these later. I'll give a lot more details on this later. The second sort of type of measures are what we call discrimination, and AUC would be an example of that. And that's asking the question, how well does the model discriminate between diseased and disease-free? Like, are we on average predicting higher probability of being diseased for the diseased group versus the non-diseased group? And again, we'll give some more details later. And the last sort of type or class we're going to be looking at is calibration. So if the model says on average there is a 5% risk of developing cancers, we would ideally want that the observed number of cancers in our data set would be around 5%. That means the model is well calibrated, at least marginally. Like, the predicted probabilities on average are close to the observed number of cases that we see in our data set. And that is what we call calibration. Okay, moving on to class predictions. So usually prediction model output predicted probabilities, prediction models for binary outcome. Like from a logistic regression, you would get you can transform the output from a logistic regression to a predicted probability of being deceased, and then obviously conditional on participant characteristics. And many measures of model performance require you to sort of, instead of having a probability, you want to label diseased or non-diseased. Rather than having 5% probability of being diseased, you want to sort of classify it into predicted class labels, diseased or non-diseased. And that requires you to select some cutoff or some threshold. There, if you fall above that threshold, you're going to say that this participant is going to be predicted to have the disease. If you fall below that threshold, you're going to be saying, or the model is going to be saying that this person does not have the disease. And this is sort of taking a continuous spectrum and making it into a binary spectrum, diseased versus not diseased. Okay. And a lot of these measures can be summarized through this 2x2 table. Specificity, sensitivity, negative and positive predictive value, and accuracy can all be sort of, that information can be obtained from this 2x2 table here, where you have the truth or the disease status here, and then you have the prediction from the model, either positive, meaning having the disease, or negative, meaning the model predicts not having the disease. So if you are in the upper left-hand corner here, that says these are participants who have the disease, and the model or the marker or the test correctly classifies them as having the disease. And the number of those people would be A here. So A means making the correct decision. They have the disease, and the model correctly tells they have the disease. If we go to the bottom left corner, we have the people who have the disease, but the model incorrectly classifies them as not having the disease. So this is making the wrong decision. This is saying people who have the disease, that's the model saying they don't have a disease. So an incorrect decision. Then moving to the upper right-hand corner, this is people who don't have the disease, but where the model says they have the disease. So again, incorrect decision, just in the opposite direction. This is basically the model saying you have a disease when you don't have it. And then in the lower right-hand corner, this is not having the disease, but the model saying you have a disease. So a correct decision. So you want to have as many people on this AD diagonal as possible, and as few on the CB diagonal as possible. Because that means you're making the correct decision the most amount of time. But not all errors are in some sense equivalent in sort of when you're making these decisions. Like incorrectly saying, or the model incorrectly classifying someone as having cancer is probably less severe than failing to detect cancer. Because you could just like, say you're doing this to an MRI, you would go through some further diagnostic workup, potentially biopsy, and that biopsy would come back negative, saying you don't have a cancer. But the sort of failure of detecting the cancer is much more severe, because you're then unable to act on the cancer early enough. And therefore maybe missing out on an opportunity to treat it, basically. So we have to be a bit, so basically not all errors here are created equals. And two very commonly used measures are sensitivity and specificity. So sensitivity is basically, among those that have the disease, what proportion does the model predict to have the disease? So now you're just looking at the group of people who have the disease, and now you want to see out of that group, how many of those did the model correctly say have the disease? Going back to this 2x2 table, we are only looking at the group that has the disease. And so the denominator here would be A plus C. And then we want to say what proportion of those does the model correctly classify to have the disease. So the numerator would be A, or all the people where have the disease and the model says that they have a disease. And specificity is sort of on the opposite spectrum of focusing on those who do not have the disease. So focusing on the group that do not have the disease, what proportion does the model say they do not have the disease? So going back to the 2x2 table, we are focusing on the group that does not have the disease. What proportion of that does the model say? does not have the disease, that would be D. So sensitivity or specificity would be, the denominator would be everyone who does not have the disease, or D plus B, and the numerator would be D. So both of these measures are between 0 and 1, and higher values indicate better performance. So ideally, in an ideal world, you would want to have both of them very close to 1. That would mean among the people who have the disease, the model said almost all of them had the disease. So it's making the correct decision in that group almost all the time. And the specificity close to 1 would be saying among the people who don't have the disease, the model is saying they don't have the disease almost all the time. So making the correct decision within that group almost all the time. Okay, and then negative and positive predictive value are just sort of moving across a different spectrum of the 2x2 table. So in sensitivity and specificity, we were conditioning on disease status. We were looking within the group of diseased or looking within the group of non-diseased. Now we are going to be flipping the tables here in the sense that we are going to be conditioning on what the model says, and we are going to be looking within the class of what the model predicts. So among those that the model predicts to have the disease, what proportion does have the disease? So going back to the 2x2 table, this was positive predictive value. So we are looking within the group of people who have it, or the test or the model says to have the disease. So the numerator would be A plus B. And then what proportion of those actually did have the disease, which is A. So positive predictive value would be A divided by A plus B. And similarly, negative predictive value is just the same thing, just looking within the group where the model predicts them to be disease-free, and what proportion is actually disease-free. So that would be D would be the denominator, that's the people who are disease-free, and the model says they are disease-free. And then the denominator would be C plus D, which is the number of people that the model says they are disease-free. And again, exactly the same sort of, we would want high positive predictive value, because that's basically saying within the people where the model predicts them to have the disease, most of them are actually diseased. And similarly for negative predictive value. So the same sort of conclusion as for sensitivity and specificity apply here. The key thing is, what are you conditioning on? Sensitivity and specificity condition on disease status. Negative and positive predictive value condition on the output or the class prediction of the model. Okay, so all these measured here relied heavily on the choice of C or the threshold. Like, do we say that everyone who has above a 5% probability, we predict them to have the disease? Or do we make that threshold 50%? That threshold determines how many the model classifies to have the disease versus non-disease. And varying that threshold is going to lead to different values of sensitivity and specificity. And that is what you're getting in an ROC curve. So an ROC curve has sensitivity on the y-axis. It has 1 minus specificity on the x-axis. And remember, and then you are plotting that for different thresholds for the class labels. So you sort of get a continuous, you get a continuous curve which corresponds to different thresholds used to say you have the disease versus you don't have the disease. And remember, sensitivity, ideally you want that as close to 1 as possible. So you want to be around sort of here for sensitivity. And specificity, you also want to have as close to 1 as possible, which means this is 1 minus sensitivity on the x-axis. So you want the x-axis to be close to 0. So ideally, if you had a perfect model, it would classify everyone correctly independently of the threshold. So you would just have a single data point here in the upper left-hand corner. But that's not going to be the case in most practical situations. You're going to be misclassifying some of them. But ideally, you want the ROC curve to be sort of as close to the upper left-hand corner as possible. So sort of the way you look at that is in some sense, you're looking for high ROC curves that are closer to the left-hand corner. So in this particular situation, the best model would be the green model here. Because for most values of C, this is going to be having sensitivity close to 1 and specificity close to 1. The second best model would be the blue model, and then you get models that are sort of less good for the other curves here. And if you had a model where you just randomly chose the labels, you would be getting on average an ROC curve that is just like the diagonal line here. Sort of you would get a straight line going from the 0, point to the 1, point. That's an indication the model has no idea what it's doing, basically. Okay, so this is an ROC curve. And some of you might have heard about the area under the curve, which is basically the area under the ROC curve. So looking at this figure, you would see the area, and that's basically taking for the green curve, that would be the area, this area here. I don't know if you can see my cursor. If not, Chris, let me know. It would be the area from this green line to here. So if you had the perfect model, it would be the area of the box here, which would be equal to 1. And an AUC, you can both think about it as the area under the ROC curve, but it also has an alternative interpretation as a discrimination variable or a measure of discrimination. So AUC measures the ability to discriminate between the disease and non-disease. And in addition to being the area under the curve, the AUC also has a somewhat complex interpretation where you basically, now you take 2 observations. You take 2 participants where one of them has the disease and the other one does not have the disease. And ideally, you would want the predicted probability for the person who has the disease to be larger than the predicted probability for the person who does not have the disease. That means you're sort of ranking those 2 people correctly. You're saying the person who has the disease has a higher probability of getting the disease than the person who does not have the disease. So that in just the setting of those 2 participants, you're making the right ranking of them. And the AUC is basically just generalizing that notion to the population level. But it's basically the proportion of pairs with one having the disease and the other one being disease-free, for whom the deceased has a higher predicted probability of having the disease than the one who is disease-free. So that's sort of its discrimination interpretation of the AUC. And this is also the area under the ROC curve. And again, this is between 0 and 1. 1 indicating your model is almost perfect at discriminating the 2 classes. 0 means you should have done exactly the opposite. It's actually exactly the opposite than if you would switch things, you would be perfect. And 0.5 means you have no idea the model has no idea what it's doing. And now the last class of measures, and I'm going in way more details about the binary measures than the continuous measures, because I think they are a little more complex in some sense. So calibration is the last class of measures that I mentioned. So they're basically asking the question of how similar are the observed number of cases and the expected number of cases. So basically, if 5% of the people have the disease, then we would like the model of average to have, if you average the predicted probabilities, we would like that to be around 5%. If that was like 60%, the model is being way more likely to say you have the disease than you actually are. But it has no effect on discrimination. So it's usually a very good idea if you're having an AUC curve or an ROC curve plus an AUC, you should usually augment that with some measure of calibration. And one way to sort of measure calibration is what we call calibration curve. There you have, and here you are sort of binning people based on the predicted probabilities. Like, so you're taking the least or the 10% of people say that has the lowest predicted probabilities, and then you're calculating the average predicted probability within that group, and you're comparing that to the number of observations who have that. And that would be this dot here. And then you do that for all the bins. And then you get a plot like this. And ideally, you want this to fall on a straight line. That would mean your predictions are on average matching the number of cases or the number of diseased people within each group. I see there is a question here. Oh, can't see the cursor. Thanks. Okay. Now I'm going to move into an example. And we are going to be using the National Lung Screening Trial as an example here. This is a large clinical trial that enrolled over 50,000 people, high-risk people for lung cancer, so older smokers or heavy smokers. And it was comparing the effect of using CTs versus chest X-ray screening for lung cancer. And this has been very influential in informing clinical guidelines, has also been used to build several prediction models, or even also been used as an external validation set for several prediction models. For those of you who know lung cancer screening, this is PLCO. It's another study that has been used to build models and has been evaluated in the NLST. And these models have varied from logistic regression models to deep learning models and so on. And the outcome here we are going to be focusing on is being diagnosed with cancer within 6 years from study entry. And this is luckily a pretty rare outcome. In a screening population, and a healthy population, but still high risk for lung cancer, we are talking about around 3% of those people, at least in this study, were diagnosed with lung cancer. And this should be lung cancer, not just cancer, within 6 years from study entry. And we are only going to be looking at the people that got diagnosed with chest X-ray, and we are going to be using a logistic regression model that we built on part of the data on a training set. So this is sort of an example of an internal validation where we have a training set where we built a logistic regression model, and then we have a holdout test set which we are evaluating that model on. Okay, so for sensitivity, specificity, positive and negative prediction value, we need a cutoff. We needed this C or the threshold. How should we select that? Many people just say, oh, you should select 0.5. Because that's like means it's like if it's below that, there's a higher chance you have you don't have lung cancer. But using that in this setting here is going to give us no information because all these are the predicted probabilities on the test set, and you see all of them are very low. All of them are beneath 20%. So if you used 50% as a cutoff, the model would just be saying no one has lung cancer, or no one will be developing lung cancer in the next 6 years. A completely useless model. But the question still remains, how should we select that cutoff? And that's a tricky question that is a very, very hard to sort of give any general advice on. Like often is select based on sort of clinical relevance of sensitivity versus specificity, what type of errors are more severe than others. You might be more willing to make false positives than false negatives or vice versa. And based on that, you might be selecting the cutoff higher or lower. But one data-driven way to do that is to use something that's called the Uden index, which is basically trying to maximize the sum of sensitivity and specificity. Or basically selecting the point on the ROC curve that is closest to this perfect model, which would be the upper left-hand corner. So you'd be selecting the threshold C that gives you the sort of the highest sum of sensitivity and specificity, or equivalently the cut point that gives you the point on the ROC curve that is closer to the upper left corner, which is where we want to be. So that's one data-driven way to select the cutoff. But obviously, this is more complex and should be studied on a case-by-case basis, depending on sort of how highly you value false positive versus false negatives and so on. And when we apply that to this data set, on the training set, data set, we got a cutoff of 0.0255, which would be somewhere like around here. So like, yeah, that was the optimal one here. So using that cutoff, we get this 2x2 table. And like we see, this is the truth. The truth is no lung cancer versus lung cancer. And here we have what the model says, predict or not predict lung cancer. And as I alluded to, lung cancer is pretty rare in this population. So we see the numbers in the lung cancer group are much smaller than the number in the no lung cancer group. Now looking at sensitivity. So sensitivity was conditional on how many within the group of people who have lung cancer, how many people actually had lung cancer. So the denominator would be 286 plus 75, and the numerator would be 286, giving us a sensitivity of 79%. And it's always a good idea to augment that with confidence intervals or some measure of uncertainty, which I did here, and there are standard ways to do that. So a relatively high sensitivity, depending on how you define high. And this is a tough classification problem. Based on the data we have at hand at the beginning of the study, it's very hard to predict who is going to get lung cancer or not. It's not like you're diagnosing cats from dogs in images or something that is a little more, that is a little easier of a problem, which is, and sort of, so if this is a good sensitivity or not has to be interpreted in the context of the problem that is being studied. And then looking at specificity, then we are conditioning on being in the no lung cancer group. So the denominator would be 5,820 plus 5,240. How many would the model predict as having no lung cancer? So that would be 5,820 divided by that. So specificity there is pretty close to 0.5. So if the model says you have no lung cancer, you're almost equally, or sorry, if you have no lung cancer, if you have no lung cancer, the model is almost equally likely to say you have no lung cancer or not. So this model is really sort of prioritizing sensitivity in some sense. It's more willing to make the error of saying someone who does not have lung cancer, the model saying it has lung cancer, then making the error of someone who has lung cancer, but the model failing to detect that. Okay, so then negative and positive predictive value are just looking at the other side of the spectrum. Basically, negative predictive value is saying among the people who don't have lung cancer, or where the model does not predict it to have lung cancer, how many of those actually did not have lung cancer? So it's 5,820 divided by 75. And in a rare case where you have an outcome that is rare, negative predictive value tends to be very high usually. Because the model is, when the model predicts you to have no lung cancer, you're very likely to not have lung cancer because the outcome is so rare to begin with. But you have the opposite problem for the positive predictive value, where the model predicts you to have lung cancer, which would be a total number of people 5,240 plus 286. Then how many of those actually had lung cancer? That was only 286. So you have a positive predictive value of 0.05. And again, pretty typical of a rare outcome situation where the denominator here can only get so high because there is just so few lung cancer in the data set. Here we have the ROC curve. We see sensitivity and 1 minus specificity on the two axes respectively. And we see we are evaluating sort of in terms of AOC, which is 0.7. And here we have the associated 95% confidence interval. So again, doing a relatively decent job, depending on how you define decent here, not far away from perfect. This is a hard task to do. And then calibration, again, we would like to see these dots here fall on a relatively straight line. And that's what we see in this specific scenario. So calibration seems to be pretty good in this case. Repeating myself here, what we are looking for here is that these dots on the calibration curve fall approximately on a straight line. That means within each risk level predicted by the model, the observed number of lung cancers is pretty close to the average predicted number of lung cancers. Okay, binary outcome. I told you I was going to spend 90% of the time on that, and I did. For continuous outcome, the mean squared error is by far the most commonly used measure, and you can actually also use that for binary outcomes. That's called the Breyer score. And the mean squared error basically estimates how far is your prediction from the observed data. So the prediction here would be the blue line. The observed data points are given by these purple circles. So for observation 1, you would take the distance between the observed value, or the purple circle, to the prediction, or the blue line. You would take the distance between that and square it. And then you would do that for every single observation and calculate the average. And that would give you the mean squared error. And that can be decomposed into bias squared plus variance. So bias, so this is a very common illustration of bias and variance. Bias basically tells you how close on average are you to the truth. Basically, if you would average all the data points, how close are they to the red circle? And variance would tell you how sort of dispersed are your data points, or not data points, your estimators. And in these situations, you see very clustered estimators. All the purple circles are very close together, and they on average are very close to the red circle. So this would mean low bias and low variance. Here we have the situations where on average they are very close to the circle, so low bias, but they are more dispersed. They are more spread out, meaning high variance. And then we would have high bias, but low variance, where you see on average they are far away from the red circle, but you're getting low variance because they are pretty clustered together. And here you have high variance and high bias, which is basically far apart and far away from the red circle on average. And the key takeaway point here is that MSE can be decomposed into bias squared plus variance. So like in order to decrease MSE, those are the two things you can focus on. And last, very briefly, for censored outcomes, these are way more complex, and that's why I didn't get into it. But there are established methods to do it, but some of them are like, it's harder to find standard software to go about this. There's the UNOS C index, which is another version of the AUC. There's time-dependent ROC curves, and there's a censored data bias curve. And here I give references that sort of discuss all of these three methods. I'm happy to answer questions afterwards, but I'm not going to go into any more details on that. There are a bunch of references. If you Google anything online, you will find probably a million blog posts on this or that about it. Here are 3 references from the statistical literature on this that I like. The Elements of Statistical Learning is a very popular book, machine learning book that has both how to do machine learning and how to evaluate it. The Introduction to Statistical Learning is a little lower level than the Elements of Statistical Learning, and then you have something that's a little more focused on diagnostic methods as the last book. And that concludes what I wanted to say. We're happy to answer any questions. I believe you are muted, Chris.
Yeah, I'm sorry, I was on mute there. Yeah, so questions either in the chat or raise hand, whichever is it's easier for people. Oh, I guess it's going to be chat because you can't raise your hands. We've tied them down. Well, while we wait, Jon, let me ask you a question. I know you didn't get a chance to talk about this, but your research has focused a lot on how these models transport to other settings. Do you want to say a few words about that?
Yeah, no, thank you, Chris, for allowing me to do that. Yeah, like Chris is saying, a lot of my sort of research interests center around the question of sort of extending evidence, generalizing or transporting these models to different correlations, somewhat related to external validation, like how do they perform in a different setting than was used for original model development. But usually I'm considering a different data setup, in the sense you might not have the outcome of interest measured in the new setting. For example, in the lung cancer screening example, you might have a cross-sectional data set, like the NHINGE or some other cross-sectional data set, where you only have covariate information available to you, but you don't have the long-term follow-up needed to obtain outcome information. Like, for example, here you need to wait for 6 years to know if they developed, if they develop cancer within 6 years. And that requires you to follow people from 6 years, which is both expensive, time-consuming, and so on. So can you do those external evaluations without having the outcome information in the new setting you're interested in doing that evaluation? So sort of asking the question of what are the conditions? Obviously, because you don't have outcome information, and the only outcome information is from the sort of the original setting, you need to have some assumption connecting the two. And then what are the assumptions? What are the appropriate estimators? How is the appropriate method of doing confidence interval construction and so on? So happy if anyone has interest in those type of settings, or even in, I would be happy to have discussions if I'm always on the lookout for data sets to apply these methods to, or these are questions that people are interested in. So happy to have that discussion with anyone on this Zoom meeting.
Yeah, I know that one problem that I'm working on that Jon is helping me with is we're developing a predictive model for dehydration in Bangladesh, but people in Africa are very interested in using it as well. And so, you know, we'd like to do a trial there, but before we do the trial, you know, how likely is it that model will actually work well? I've also worked with kidney prediction models where, you know, we develop the model in one population, let's say in the US or Europe, where we have a lot of data, but they want to use it in other parts of the world. And how well is the model going to work there? So that's, I think, a really important question. We have there's a question in the chat, if you want to take a look at that.
Oh yeah. So the question is basically, I presented methods for the chest X-ray arm, but how well does it do for the CT arm? So I mean, the CT arm is really the arm of interest here. CT, and the NLST was probably the main driver of moving the NLST. The results from NLST basically showed very strong reduction in lung cancer specific mortality was associated with CT screening, around 20%. So that moved the sort of standard of care to CT screening. So CT screening is probably the arm of interest here. I sort of arbitrarily, and because you can externally validate it in the PLCO, I chose the chest X-ray arm. But if I did this for the CT screening arm as well, and the results were honestly pretty similar. And that's more of a function of, this is just a pretty hard problem in general. Like our ability to distinguish between, based on sort of baseline data, whether someone is going to be diagnosed with lung cancer within 6 years is just pretty limited. So the signal to noise ratio on the data is rather low here, is sort of what I'm trying to say. And that applies somewhat almost equivalently to the CT arm versus the chest X-ray arm. Slightly less so to the CT arm, but still the problems, the fundamental problem why these evaluation measures are not all stellar ones is the fact that this is just a hard classification or prediction problem. Does that answer your question, James?
Yes.
Oh, yeah, thanks.
Yeah, and I was, looking at this 2x2 table here. You mentioned the 0.5 cutoff, which is kind of used as a default in a lot of programs, and you mentioned, of course, that would predict just about everybody to have no lung cancer. I was wondering if you could say a few words about some of the other measures that are used in the machine learning community. So, for example, they use accuracy and F1 and these kinds of things, and how those relate here. And accuracy in particular, if you just predicted everybody to have no lung cancer, since almost nobody has lung cancer here, you'd get an almost perfect prediction. So,
yeah, the reason I did not discuss accuracy is I don't super like it. So accuracy is basically just what proportion so accuracy is defined out of the total group of people, what percent were correctly classified. So going to the 2x2 table, this would be, the numerator would be A plus D, and the denominator would be A plus B plus C plus D. So basically, how often are we doing the correct classification? In this, the reason this I don't think is a useful measure in this lung cancer screening example, for example, is a model that just says no one has lung cancer is going to do very well on that measure, because you would be getting the denominator to be 5820 plus 5240, and so that would be the numerator, and the denominator would just be that adding those tiny number of lung cancer cases. So I'm pretty sure, I haven't done the calculations here, but if you were trying to optimize the performance of this model in terms of accuracy, the optimal cutoff would just to say no one has lung cancer. And that model is penalizing false positive and false negatives equivalently. It's basically saying it's as bad of a decision to failing to detect or to say someone is at high risk for lung cancer versus failing to say that, or failing to say someone at a high risk for lung cancer will actually develop lung cancer versus saying someone is at high risk for lung cancer, but they did not develop lung cancer. So I think it's, at least in this setting, and this applies to a lot of health data, is that I think it's more informative to look within groups of. That's what sensitivity and specificity do. They look within groups of diseased and non-diseased, and negative and positive predictive value look within predictions of the model. And I think that's somewhat more informative here. Like this would be a pretty bad cutoff for accuracy. We have a pretty low accuracy here because we are making so many incorrect decisions here, where we are saying the model has the model predicts lung cancer, but it does not have lung cancer. We have 5 to 40 of those.
Mm-hmm.
So yeah, I don't like accuracy is a simple answer here, but you see it all over the place, and it makes most sense where there is where you can put equal weight on the two. On the two types of errors.
Mm-hmm.
What about likelihood ratios for low frequency? Sorry, can you define likelihood ratio? Like, are you talking about like two models and when you're comparing the likelihood ratio between the two? Like you have a logistic regression model, you have a likelihood ratio associated with that.
Yeah, or maybe the diagnostic test criterion, which is kind of if you take out the prior rates.
Like can you repeat if you take out the prior?
Right. I mean, the prior rate would just be the prevalence, right? So
yeah.
If you take that out, it's like what information are you gaining from the model, I think is the question, really.
Oh, I see. Okay, yeah. Yeah, I see those less used in the binary setup, but you definitely see it more in the continuous setup, but even the time to event outcome setup, where you would like, this applies both to likelihood ratios and MSE, like how much is the MSE decreased when you are at this variable to the model, or how much does my likelihood ratio increase when I add this variable to the model? This has a close relationship to R squared in like which you've often seen in the regression context, which I believe is like the sum of squares of the model, or the sum of squares of the total sum of squares sum of squares of the model divided by the total sum of squares. How well do you do within the model, and how compared to how well do you do if you only had an intercept, no model? And this is very related to those type of measures. So I think they are more used in the continuous case. And the nice thing about these is you can translate them to an R squared type of a measure that sort of, if I told you an MSE decreases by 5, I have no sense. Is that a good decrease or a bad decrease? That could be, and that is heavily sort of dependent on the scale of the outcome. But if I told, so this R squared type of a thing is a way to standardize it, sort of force it to be between 0 and 1. And that gives you a better sense if 5% increase is good or bad. Yeah. And this, I think similar principles could probably be applied to any measures of model performance, AIC, BIC, likelihood ratio, like, or not model performance. This is maybe more model fit rather than performance.
I think also there's also the issue of just, if you take the ratio of the probability of disease for positive test to no disease for positive test, right? You can use Bayes' rule to rewrite that in terms of the prior ratio times the likelihood ratio, and I think that might be also what's being referred to here. So it's basically the probability of a positive test given disease to the probability of a negative test given disease, and that's another measure that sometimes people will use, just sort of flipping things around. But okay, it looks like we are at 1 o'clock. Thank you very much to Jon, and thank you to all of you for attending. And there's been some messages in the chat about recordings and so forth, and we'll look forward to seeing you all again in the spring, or maybe the late winter when we pick up again with another couple of talks. So thanks again for attending.
Yeah, thanks everyone. Thanks, Chris.
Bye.
Bye-bye.